Hello, my name is Kyung-Hwa Ashcraft. I will present environmental exposure to mercury from fish consumption in Madre de Dios, Peru. 1.0 Introduction. Gold mining in the Peruvian Amazon has led to mercury contamination and public health concerns. Mercury contamination is formed by bacteria in waterways, which accumulates in fish, our primary food source. The purpose of this presentation is to identify treatment solutions to mercury exposure and fish consumption when humans are exposed to the metal through a dietary source. 1.1 Table of Content, Purpose, and Objectives. In Madre de Dios, Peru, mercury poisoning is by understanding the mechanism by which mercury binds to proteins in fish tissues. One of the objectives of the study is to focus on the treatment solutions to human environmental mercury exposure and fish consumption when humans are exposed to the metal through a dietary source. Finally, by analyzing mercury binding to human brain tissues, this study proposes new chemical treatments for mercury exposure. Methylmercury enter the cell membrane. Many proteins transport Hg to mercury, 2, reductase. Hg, 2, is reduced to Hg, 0, within cytoplasmic compartments by mercuric reductase in bacteria and archaea. For instance, Merp, which is a paraplasmic mercury transport protein, is largely found in gram-negative bacteria and transports Hg through from the outer membrane into the inner membrane, then holds the Hg for another protein to bind to it before transporting it to mercury, 2, reductase. Merp has the metal binding domain motif, GMTCXXC, which is a highly conserved region of functional importance as shown in Figure 6 below. Mercuric ion transport systems in bacteria facilitate the uptake of Hg, 2, salts and their deliver to the mercury, 2, re-educate active sites. Examples of these transporters include Merck, MERF, and MERT with different sets of topologies, Yamaguchi et al., 2007. The paraplasmic proteins Merp and MERT interact with transporters, Friedman et al., 2012. 2.0 Mercury binding to proteins in fish tissues. Methylmercury binds tightly to fish protein following absorption through the gills or when fish eat contaminated sources of food. In all fish tissues and muscles, mercury is often rightly bound to proteins. 2.1 Mercury binding to proteins in fish tissues. Studies on salmon and tuna fish muscles have shown that beta-actin is a novel mercury-binding protein, which could also bind silver, Ag, bismuth, Bi, and copper, Cu. It was also found that mercury bound to this protein accounts for about 30.2 to 37.6% of the total mercury in fish muscles, Known et al., 2021. In edible fish, proteins are the main mercury accumulation sites, and this increases human exposure to mercury after gastrointestinal digestion. 3.0 How mercury binds to human brain tissues. In the bloodstream, MHG binds to sulfhydryl groups, particularly cysteine, and is distributed to peripheral tissues by one or more transporters, such as the cysteine transporter. As shown in Figure 2. As shown in Figure 3, organic and inorganic forms of mercury pass the blood-brain barrier and enter brain tissues, where they become potent neurotoxins. As well as being lipophilic, mercury can easily penetrate various organs of the body, including the brain, without toxic effects. The human brain contains two kinds of mercury, methylmercury cysteiniate, CH3HG-SCH2, that can be toxic, and mercuric selenide, HGSE, that is inert and non-toxic. As a result of mercury's affinity for selenium and demethylation in the central nervous system, mercury poisoning can be delayed. 3.1 How mercury binds to human brain tissues. In humans, mercury can bind to thiol, which is a single sulfur group containing molecules such as cysteine as shown in Figure 8 below. this binding allows Hg to be transported across the blood-brain barrier. The ability of mercury to cross the blood-brain barrier has become a major concern. One cure, acid. Study of mercury redox reactions in the extraction of organic acids from vegetation roots. The role of citric, succinic, and tartaric acids in metal redox reactions was investigated in the laboratory. Results showed that tartaric acid significantly stimulated mercury reduction, while citric acid inhibited it. Nevertheless, succinic acid improved reduction rates at low concentrations while inhibiting the process at high concentrations. Among tartaric acid, succinic acid, and citric acid has the highest reduction potential. In terms of mercury activation, the three acids follow the following trend: citric acid, tartaric acid, oxalic acid. In humans, mercury and lead are among the most toxic biometals, and acids have been shown to detoxify them. Based on the table 3 below, ascorbic acid, GBII, had the lowest blood lead concentration. Studies show that these acids have the potential to address mercury toxicity in the environment and in the human body, providing insight into developing new pharmacotherapies. 2. Cure, pectin. 1. Mercury destroys the structures of proteins. Mercury binds strongly to selenocysteine, and cysteine amino acids react with CH3Hg+, because of their S bonds as shown in equation I below. 2. This reaction occurs when cysteine or other amino acids related to cysteine transport CH3Hg+. 3. Pectin is a degalacturonic acid-based heteropolysaccharide in plant cell walls. The pectin can dissolve a denatured protein, RCH, NH2, COOH, or the complex formed when mercury binds to proteins. 2.1 Pectin molecule. Pectin interacts with a wide range of molecules and has been used to remove toxicity via therapeutic genes, molecules, and cells. Summary. I present the new treatments when human exposure to mercury from dietary sources. By analyzing mercury binding to brain tissue, this study proposes new chemical treatments for mercury poisoning. In this presentation, acid and pectin are discussed as the two main treatment options.